This is a video to help you through the activity on the central dogma of protein synthesis. The central dogma is the understanding of how DNA is converted into messenger RNA and then converted into protein. It's the understanding of how our DNA is used to make proteins of specific sequence based on the sequence of the DNA. It's helpful to think of these things as different languages. DNA and RNA are in the language of nucleic acids, while protein is in the language of amino acids. Proteins are polypeptides. They are polymers of amino acids, which have a peptide backbone and side chains hanging off of the backbone. The sequence of a protein is absolutely essential for its function. Each protein has its own specific sequence, and the sequence of an amino acid chain, or the sequence of a protein, originates from the sequence of DNA. DNA are the blueprints for your cells to make proteins of specific sequence. Recall that DNA is a double helix, two strands of DNA that are complementary. They interact through hydrogen bonding, and each strand has a complementary sequence to the other. We'll see in this activity that the sequence of DNA allows our cells to make specific proteins based on that sequence. This is the central dogma of protein synthesis. It's an understanding of how we convert DNA to messenger RNA into proteins through processes called transcription and translation. Remember that DNA and RNA are in the same sort of language, maybe different dialects of the same language. And converting from one to the other only requires transcription. Converting from the nucleotide language to an amino acid language, like in the conversion of RNA to proteins, takes a translator, someone who speaks both languages. This process of converting the RNA sequence into a protein sequence is called translation. Here's a cartoon that specifies where transcription and where translation are happening and also shows some of the other molecules involved. This cartoon is showing a picture of a cell. This area is the cytoplasm, and this is the nucleus. Inside the nucleus, DNA is transcribed to messenger RNA by RNA polymerase. RNA polymerase is a catalyst that creates the messenger RNA from the DNA template. Once the mRNA is formed, it moves out of the nucleus into the cytoplasm, where it is translated into a protein sequence by something called the ribosome. The ribosome contains RNA and protein, and it acts as a catalyst to use the RNA template to make a protein. Pause the video now and answer the questions that are underneath this figure in your activity. Let's take a closer look at transcription. This is a cartoon showing a DNA sequence. The double-stranded DNA is represented by the dark blue and light blue parts of this figure. Notice that the dark blue part has a complementary sequence to the light blue part. When DNA is transcribed into messenger RNA, it is first unzipped. The hydrogen bonding between the base pairs of complementary sequences are broken so that another type of nucleotide can come in and make a new complementary sequence to the original template DNA. In this figure, the pink sequence represents messenger RNA that has been created from the template DNA strand. Let's carry out this process based on a specific sequence of DNA. If this is your template DNA sequence, write the complementary messenger RNA sequence to this template DNA. Pause the video now and try this. If I start at the 5' end of my DNA, when I make the RNA sequence, I still want to start in an anti-parallel fashion, just as if I were writing the complementary DNA sequence. So I would start at the 5' end, and I would write a complementary sequence of base pairs. Except for messenger RNA, I do not have T, I only have U. So for every A in the DNA sequence, I write a U. For every C, I write a G. For every G, I write a C. And for every T, I write an A. This is the complementary messenger RNA sequence for this DNA template. There's a helpful video on transcription from the DNA Learning Center. If you click on the link below, it will pause this video and take you to the transcription video. Please do this now. What we just discussed was transcription. This is a process that happens with the catalyst RNA polymerase. This is a catalyst enzyme, and it uses a DNA template to create a messenger RNA sequence that's complementary to the DNA. Once the messenger RNA is created, it leaves the nucleus and it takes part in translation, which uses the template of the messenger RNA to make a protein sequence. This requires translation, and it needs a translator. This is the mRNA codon chart from your textbook, and this is the translator. You'll see where these things come from in a moment, but first notice that there are 3-letter codons. The sequence of messenger RNA is broken down into 3-letter groups that code for specific amino acids. Notice that this chart is organized according to the first 2 letters of the codon. The first letter is listed here, and the second letter is listed here. If I wanted to find a sequence AUG, since the first letter is A, I would just come down here, U is here, and then I can find AUG, which is methionine, or the start codon. All of these codons are read from the 5' to 3' end. We'll address this again in a moment. What is the meaning of these codons? The codons are matched with an anticodon on a piece of transfer RNA. This is another figure from your textbook, and it shows what is going on in the central dogma. Let's review what we already talked about. First, messenger RNA is made on the DNA template from RNA polymerase. In this figure, the DNA template is the purple double helix, which is being unzipped by RNA polymerase, and the template DNA is being used to make a piece of messenger RNA. The messenger RNA is shown in blue. Once the messenger RNA is created, it leaves the nucleus. Here's the messenger RNA outside of the nucleus. It meets the ribosome, which is shown here in the purple and dark blue cartoon. This whole thing is the ribosome, and the ribosome moves along the piece of messenger RNA from the 5' end to the 3' end. In this figure, it's shown that the ribosome is moving from the left to the right. So the left-hand end here must be the 5' end, and the end over here must be the 3' end. The sequence of the mRNA bases is specific. It carries the codons. Here's the codon GCC. And if you look at your codon table, you'll see that GCC codes for the alanine amino acid. AUA is a codon that codes for isoleucine. And then you can find the next amino acid in the sequence by looking up GUC, which is the next codon. The messenger RNA hydrogen bonds to the anticodon on transfer RNA. Let's take a closer look at what's happening right here. Here's the ribosome, here's the messenger RNA, and here is the transfer RNA for isoleucine. This cartoon is showing a piece of RNA which contains an anticodon, or the complementary sequence to the codon AUA. This is the transfer RNA for isoleucine. It contains the anticodon for isoleucine, and it is bound to the isoleucine amino acid. There are 3 types of RNA that take part in translation. Messenger RNA is the messenger that carries a message from the nucleus to the cytoplasm. The ribosome itself is actually made of some RNA, and the ribosomal RNA is called rRNA. The third type is transfer RNA. Transfer RNA contains the anticodon and the amino acid. This is the translator between the codon sequence and the amino acid that is specific for that codon. Here's another picture of transfer RNA. Here we have the same figure that we saw here, and this is a more detailed figure of what the tRNA is. Here the 3' end is shown with just the hydroxide. This is where the amino acid would be attached. In the figure over here, this would be the 3' end, and the ball is representing an amino acid. RNA, unlike DNA, does not have to be in a double helix. RNA can form other structures. This is showing the sequence of an RNA with a loop, and another loop, and the hydrogen bonding between base pairs is shown with the dotted lines. Notice that this tRNA forms a specific shape, but is not a double helix. On one of the loops is the anticodon, and on the 3' end will be the amino acid that's specific for this anticodon. This anticodon would be read from the 5' to 3' end as CGU. Therefore, its codon would be ACG. Here's another picture from a different textbook. This picture shows the space filling model. And in this particular picture, the whole thing is flipped upside down compared to the last one I just showed you. In this one, the anticodon is shown at the top, and the 3' end is shown down here at the bottom with an amino acid attached. This amino acid is phenylalanine. Its anticodon is GAA, and so its codon would be UUC. Look up UUC in your codon table and confirm that it codes for the amino acid phenylalanine. Here is a space-filling model of what this piece of tRNA would look like. And every piece of tRNA looks very similar. It has an anticodon on one end of this bent molecule. And the acceptor stem, or the 3' end here, is where the amino acid is bound. Here's the amino acid. I'm circling it with my cursor. Here's another cartoon to show what happens. Here's your RNA template in blue. And the anticodon on each tRNA is complementary for the codon on the messenger RNA. Each anticodon is attached through the piece of tRNA to an amino acid. Here's an anticodon and its amino acid, an anticodon and its amino acid. In this cartoon, this whole pitchfork-shaped thing represents the tRNA. Here's a closer look again. So the things I've boxed here represent the same thing. Here you have the anticodon, and here's the amino acid, and they are connected by the tRNA. The tRNA is what translates the codon sequence into an amino acid sequence. The ribosome does the job of making bonds between the amino acids that are lined up along the chain here. So it would take the nitrogen here on this amino acid and make a new bond to the carbonyl. This would make a new nitrogen-carbon bond, or a peptide bond. This happens as the ribosome moves down the sequence from the 5' to the 3' end of the messenger RNA. This codon chart must be read in the 5' to 3' position of the mRNA. You could make another codon chart that could be read by the anticodons, or you could make another codon chart that would be read in an opposite direction. But it is convention to always read codons from the 5' to 3' end. This is convenient because if you translate an mRNA sequence from 5' to 3', you automatically put the amino acid sequence from the N terminal on the left to the C terminal on the right. And so you are consistent with the conventions used in both cases, 5 to 3 for RNA and DNA, and N to C in proteins. Here's the figure again, and understanding this figure is important to your success. Read through each one of these steps and make sure you can understand what's going on here. The best way to test whether you understand is to recreate this sketch or write a paragraph that explains each step of the process. There are videos available to help you understand this. The transcription video link is here, and the translation video is here as well. Stop now and watch these videos several times to make sure that you can understand the transcription and translation process. This is the central dogma of protein synthesis from a DNA template. Before we end, I'd like to mention that DNA replication is another process that happens with DNA. DNA replication is represented by this cartoon. It involves proteins and enzymes, which are not shown here, but this cartoon represents the overall process. You have your original double-stranded DNA in the cell nucleus. When the cell wants to divide, it must copy its DNA, and it does so by unzipping the DNA into single-stranded DNA. The protein that does this is called DNA helicase. So unzipping happens through DNA helicase. And then DNA polymerase will come along and it will place a nucleotide complementary to the original strand. This figure is showing the new strand of DNA that is being created from the template or the parent strand. Each one of the original strands serves as a template to make two new double-stranded DNAs. During this process, mistakes can happen. Let's say one of the proteins accidentally put a T here instead of a G. There would be a mistake and a mismatch in the DNA base pair. This happens about 1 in every 100,000 base pairs. There can be quite a few mistakes since our genome is made out of more than 3 billion base pairs. There are proteins in our cells whose specific function is to find these mistakes and fix them. And so most of the time when our cells divide, We make an identical copy of our original DNA. But sometimes a mistake will slip by. This can happen if there is a problem with the proteins that replicate DNA or a problem with the proteins that are checking the mistakes. Or it can just happen once in a while because of chance. When a mistake happens and it is transferred to a new cell, the mistake is a mutation. There are several different types of mutations that are described in your activity. Please read the information that's provided and answer the questions, and check the key to make sure you're correct. To be clear, DNA replication is not part of the central dogma, but it is something that involves DNA. The central dogma is the conversion of DNA into protein. Thanks for watching this video on the central dogma of protein synthesis. If you have any questions about this video, feel free to post them in the comments. And if this video is helpful for your learning, please let me know by giving me a vote.